Morning, ladies and gentlemen. Jean-Louis, thank you very much for this kind invitation. So when Jean-Louis was asking for suggestions for this conference here in Rome, I did propose to have a talk on biomarkers, and I was expecting that Jean-Louis would select somebody who is an expert in this field and who has published on biomarkers. He actually asked me to give this talk, and I have not personally been studying this in my research, but I'll do my best to speak about this very, very interesting topic. So in a way, we could say that everything that we're talking about here in Rome is really a biomarker. What is a biomarker? It is really a marker of some type of biological activity, which could be physiological or pathological. And all of these different monitoring techniques that we are discussing in a way relate to a form of biomarking. The question that we should ask ourselves is, what are we expecting from a biomarker? What specifically are we marking? What process or what type of cell, what type of tissue? I think it's worthwhile to ask ourselves, what are the characteristics of the ideal biomarker? And needless to say, there are none of the molecules that I'll be speaking about today represent any of these ideal characteristics in their sum. And finally, I'll talk a little bit about the issue of assessing and validating biomarkers and determining their usefulness in clinical practice. So what do we expect from a biomarker? I think we expect that it's going to help us to detect disease, usually disease that is clinically latent or occult. We will expect that it's going to help us to quantify the disease severity or the extent of a lesion. We increasingly are expecting that biomarkers will tell us something about the underlying biology of a disease, that it might let us track the natural history of a condition. One of the most compelling or important reasons that we are looking for biomarkers is that we are looking for prognostic markers. And finally, we are increasingly beginning to use biomarkers as ways to determine the effectiveness of a treatment. So in a very simple view, we could say that we have these different molecular constituents of the brain. They can be either cellular, they can be extracellular, which in the presence of a neuropathological process will lead to a biomarker that becomes detectable in some kind of biological fluid that is outside the brain. And so this could be the blood, the CSF, it could be the urine, it could be some sort of interstitial tissue. But I think this view is maybe a little bit simplistic. If we think about an acute neurological disorder, we know that there is a tissue response to that disorder, and that tissue response will be one of the major determinants of the ultimate clinical outcome and phenotype that we observe in the long term. However, I think it's increasingly clear as well that the tissue response is not only an issue of injury, but it's also involving very complex reparative, regenerative mechanisms that are either due to compensation or to generation of new cells. And this occurs in the brain in a very vigorous fashion. We also know that with many acute neurological disorders, in particular traumatic brain injury and also cardiac arrest, there is a very intense process of neurodegeneration that occurs over the long term, and the underlying mechanisms of such are not really very well understood. Finally, I think it's important to understand the role that neurologic reserve plays in determining our overall response to injury and also in the repair and regenerative process that occurs after injury. And when I talk about neurological reserve, I'm talking about the innate or intrinsic ability of an individual to respond to an injury, to a neurological injury, which may be extremely variable and may be one of the principal drivers of outcome. And finally, last but not least, I think it's important to remember the genetic sources of variance, which are probably extremely important and underappreciated in terms of understanding outcomes after acute neurological disease. And so all of these steps in this process that I'm describing to you here are potentially amenable to biomarker analysis. Really, much of our focus in terms of biomarkers has been on describing and understanding and tracking the acute injury process. But I think in the future we're going to see more and more biomarkers that are directed towards understanding the reparative, the restorative process that occurs after injury, towards understanding the genetic sources of variance, towards understanding the neurological and cognitive reserve that is such an important determinant of outcome. So in the largest sense, we can think of, again, many different techniques as biomarkers, electrophysiological techniques, which we will discuss later today. Neuroimaging can be thought of as a form of biomarker. Today, for this presentation, I'm going to focus on biomarkers that are detectable in biological tissues, in particular within the brain, for example, by measuring levels of molecules in the brain interstitial fluid using microdialysis, within the blood, or within the CSF. So what are some of these molecular constituents of the brain which might be the targets or the sources of the biomarkers that we're measuring? Well, of course, there's the neuron that we're interested in, but there's so many other types of cells within the brain. There are astrocytes, oligodendrocytes, microglia, the cells that line the ventricles, the ependymal cells. There are the cells that compose the vasculature, the endothelial cells, the smooth muscle cells, pericytes. All of these are potentially sources of biomarker molecules. And then, of course, there's a host of molecules that are associated with the extracellular matrix, which increasingly are being seen now as well as potential candidate biomarkers. So we talked about the molecular constituents of the brain, which may form the source of a biomarker, but this has to occur in association with a pathophysiological process. And what are some of the processes that can lead to the apparition of a biomarker within the bloodstream, for example? So one of them has to be blood-brain barrier disruption, which is of course very common in acute brain disorders, although it's not uniform, and some brain disorders are associated with only very transient or minimal blood-brain barrier dysfunction. There's of course neuroinflammation, there's release of excitotoxic molecules, calcium-mediated toxicity, oxidative stress, mitochondrial failure, disruption of the cytoskeleton, cell death, which can be either apoptotic or necrotic, and then there's the long-term neurodegenerative processes that occur. So all of these processes potentially could be very interesting in terms of measuring biomarkers. And then there's, of course, markers of tissue repair and regeneration. Interestingly, many of the inflammatory signaling molecules that we associate with tissue injury and tissue destruction, molecules like interleukin 6 or tumor necrosis factor alpha, are actually intensely involved in the regeneration of tissues as well, and are very important in terms of understanding how the cells and axons are reconstituted in the setting of acute injury. There are molecules that have been associated with axonal regrowth. There are molecules associated with synaptogenesis, with angiogenesis, and molecules that have been linked to stem cell biology. Differentiation and migration of stem cells are all associated with specific molecules that are potentially measurable either within the brain or in the bloodstream. So what are the characteristics of the ideal biomarker? I think it can be summarized with these 10 points. It has to, of course, be something that is quantifiable. The biomarker needs to identify a specific brain site or process, not necessarily a disease. There should be a correlation between disease severity or lesion size and the biomarker level. Ideally, there should be a correlation with outcome. We want our biomarker to be both specific to the brain and specific to the disease. We also want it to be very sensitive. We want it to be reliable. We want the plasma kinetics to be amenable or clinically relevant. So for example, if you have a biomarker whose level goes up 3 weeks after injury, that's not going to be very helpful to any of us. If you have a biomarker whose level goes up for 20 seconds and then goes down again very rapidly, that's not going to be helpful. So you need to have appropriate and stable plasma kinetics. You want the labs to be able to turn around your biomarker rapidly. It's no use to have to send your biomarker to the Mayo Clinic and wait for 2 weeks for the result to come back. And you want it to be cost effective. It's no use to have biomarkers that are exceedingly esoteric and expensive. So what are the different approaches to measuring biomarkers? We can either measure a single marker, we can measure a peptide, a small molecule, we can measure levels of expression of specific genes, both mRNA or we can look at panels of biomarkers, and finally, and perhaps most interestingly, we can look at expression profiles, and this can be expression of genes using, for example, microarrays, or expression of proteins using proteomic approaches. We can look at expressions of different types of lipids, a new field which is now being referred to as lipidomics, and we can look at different constituents of the cell metabolism, and this is sometimes referred to as metabolomics. So in the classic view, when we think about a biomarker, we start with an intuition, with a hypothesis. We think this molecule, A, may be relevant to acute brain disease, and then we go about measuring its levels in association with a specific disease process. So this is the sort of classic, traditional method to implement or to validate a biomarker. But increasingly now, given the multiplicity of molecular responses to acute brain injury, we're relying on these expression profiles to actually help us select which are the biomarkers that are going to be relevant. So you can use a gene array, for example, which is going to tell you about the expression of dozens or hundreds potentially of different molecules within the setting of an acute neurological process. And then by looking at those molecules that are most expressed, you can then test them individually or collectively to determine their relationship to an acute neurological disease. When we think about the trajectory that a molecule has to take when it goes from the brain to, let's say, the bloodstream or the CSF, there are multiple barriers that those molecules have to traverse. So initially, if we're talking about intracellular molecules or cell membrane-bound molecules, they have to actually leave the cell. They have to go through the cell membrane, and they have to enter into the interstitial fluid. But that's just the first step. Subsequent to that, the marker will have to get into the bloodstream by going across the blood-brain barrier, or if we're talking about the CSF, it'll have to go through the brain-CSF barrier. Some molecules are exchanging freely or not so freely between the blood and the CSF, and there is the so-called blood-CSF barrier that's going to be involved. So the reason why I'm showing you this slide is because there are multiple barriers which need to break down in order for a biomarker to become apparent and detectable and measurable within the fluids that we're looking in. So what are some of the important neurological biomarkers? So there are those markers that have been primarily associated with neurons. The most commonly cited one is this neuron-specific enolase. There's also the tau protein, amyloid beta. There's brain-derived neurotrophic factor. And then there are some more recently characterized proteins that I'll talk about very briefly. There's the heavy chain neurofilament. There is the ubiquitin carboxy terminal hydrolase. There is breakdown products of spectrin. And then there's a number of different markers that have been associated primarily with astroglial function, and this includes most commonly cited one is S100 beta. There's also GFAP, glial fibrillary acidic protein. There is myelin basic protein, and then a number of others, basic fibroblast growth factor, glial-derived neurotrophic factor, nerve growth factor, etc. And then there's a host of other markers that have been linked primarily to microglial function and dysfunction, and which have been very poorly studied, at least in the clinical realm. So let's talk a little bit about the three most commonly discussed biomarkers: S100 beta, GFAP, and neuron-specific enolase. If we look at S100 beta, the source, as I mentioned, is primarily astrocytes and also oligodendrocytes. It can be measured in the blood, it can be measured in the CSF. It has been validated in a number of different clinical settings, both in terms of its diagnostic and also its prognostic importance: ischemic stroke, subarachnoid hemorrhage, traumatic brain injury, cardiac arrest. What about glial fibrillary acidic protein? It's also primarily produced in astrocytes. It also has been identified in ependymal cells. It can be measured in the blood and in the CSF, and it has been validated primarily for ischemic stroke, traumatic brain injury, to a lesser extent in cardiac arrest and subarachnoid hemorrhage. And then finally, neuron-specific enolase. which is believed to be more specific to neurons themselves, as its name suggests. It's measurable in blood and CSF as well. It has been validated as well in cardiac arrest, ischemic stroke, and in traumatic brain injury. This is a very recent paper looking at the relationship between neuron-specific enolase levels and markers, independent markers of brain injury. So here you can see on the left-hand panel the volume of brain damage in cardiac arrest as assessed using diffusion weighted imaging. And you can see that there is a correlation between the volume of that damage and the neuron-specific enolase levels as measured in the blood. On the right-hand side of your panel, you can see in those patients with cardiac arrest who died, when they looked at the burden of brain damage as detected using neuropathological specimens, you can see that there was also a relationship between the pre-mortem NSC levels and the post-mortem evidence of brain damage. So there is some internal validation here of NSC as a marker of brain, of the extent of brain damage. This is a paper going back 9 years, demonstrating how serum 100 beta levels are extremely elevated in the setting of both cardiac arrest and traumatic brain injury. You can see that when compared with controls, the levels increase 5 to 10-fold. You can see that these levels tend to be extremely high at the outset and that they tend to decline over time, although they remain elevated even 12 hours after the insult. This is perhaps the largest validating study of biomarkers in cardiac arrest, paper from Zandbergen in the Netherlands. And you can see that they are comparing the receiver operating characteristic curves of both NSC and S100 beta in cardiac arrest victims. And in terms of their ability to predict poor outcomes defined here as death or vegetative state at 1 month. And you can see that the area under this receiver operating characteristic curve was indeed larger for NSC than it was for S100 beta in this large cardiac arrest cohort. And largely based on this particular study, the American Academy of Neurology actually recommends the measurement of NSC as being potentially a very useful indicator of prognosis for patients after cardiac arrest. Specifically, if your NSC level is greater than 33 micrograms per liter, that has been associated with an extremely high rate of poor outcomes. And so it can be used independently as a marker in patients after cardiac arrest. Now, I should qualify this statement by saying that these guidelines from the American Academy of Neurology were largely drafted in a period when therapeutic hypothermia was not being used in a broad sense, and that it is possible that with the advent of therapeutic hypothermia, in fact, there are data showing that the value, the predictive value of NSC after cardiac arrest in patients who have been receiving therapeutic hypothermia may be less. So, let me talk a little bit about severe traumatic brain injury. And here you can see that there is a close relationship between S100 beta levels and short-term mortality. You can see that the higher the levels, the lower the survival over a 30-day period in this cohort of severe traumatic brain injury victims. Here you can see the relationship between S100 beta and GFAP and 3-month outcomes. And you can see that there is a very nice separation between those subjects who have poor outcome and those who have good outcome for GFAP, a much better discrimination than occurs with S100 beta. Here you can see that if you combine an assessment in this very recent paper of TBI subjects, if you combine the assessment of GFAP with S100 beta, you can see that when both levels are very high, the proportion of patients who are dead at 6 months is extremely high. And the proportion as well of patients with poor outcomes is going to be extremely high. So this slide illustrates this concept that potentially measurement of a single biomarker is not going to be sufficient, but that measurement of multiple biomarkers is indeed going to be extremely helpful. Here you can see in this paper from a Dutch group, the receiver operating characteristic curve comparing 3 different biomarkers in patients with traumatic brain injury. And you can see if you look at the 6-month Glasgow outcome scale, the 2 molecules which tended to have the largest area under the curve were GFAP and neuron-specific NLAs. They tended to be much more predictive for traumatic brain injury patients than S100 beta was. And here you can see in the setting of subarachnoid hemorrhage, the proportion of patients or the distribution of patient outcomes at 1 year using the Glasgow outcome scale. And you can see that those patients who had the highest S100 beta levels were the ones who tended to have the worst distribution of outcomes at 1 year. This is a very interesting paper also coming from the same group in Paris of Louis Pubasset. And you can see that when they implemented the systematic use of HMG-CoA reductase inhibitors of statins, they found, interestingly, that the S100 beta levels were consistently lower in those patients who received statins. So they're not able to show any outcome difference when they use statins in patients with subarachnoid hemorrhage. However, they did see that there were lower levels of S100 beta in this patient population. Let me talk to you a little bit about this protein, which I think is, we're going to hear a lot of discussion about, which is a protein called tau. Tau is a protein that stabilizes the microtubules. And normally, the presence of tau is going to be extremely helpful in terms of allowing transport along the microtubules in the axons. Now, when tau becomes hyperphosphorylated, it's going to disassociate from the microtubules, and this is going to impair axonal transport, and it's going to impair neuronal function. Now, we know that tau has been linked tau proteins can aggregate, and their accumulation leads to neurofibrillary tangles, which has been associated with a number of different neurodegenerative disorders. Now, it turns out that you can measure tau levels in the blood and also in the CSF. This is a very interesting study, 39 patients with severe traumatic brain injury, and they found that when you measured tau levels in the CSF, you could, on between days 2 and day 6, there was a significant discrimination between those subjects who had a poor versus a good 1-year outcome. So tau protein is one of these proteins that I'm quite certain you're going to be hearing a good deal about. And then just the last protein that I'm going to discuss, and then I'll be done, and we can have questions. And this is amyloid beta. So amyloid, we all know about amyloid beta, which has been associated with the neurodegenerative disorders such as Alzheimer's disease. It's the main constituents of amyloid plaque. We know that amyloid precursor protein undergoes cleavage to produce this protein amyloid beta 40 and amyloid beta 42. Now, it turns out that you can actually measure levels of amyloid beta in the interstitium of the brain. And this is a very nice study from a group in which Nino was participating, where they measured levels of amyloid beta over time in subjects with severe traumatic brain injury and also with subarachnoid hemorrhage. And they found that these levels were extremely high and tended to go up with time. They also found a very interesting relationship between amyloid beta levels and level of consciousness. So those individuals who had a higher level of consciousness actually had higher interstitial levels of amyloid beta. So this was obtained using microdialysis. So now I will skip to my conclusion. So to summarize, so biomarkers of acute neurological disease can be accessed via the blood, via the CSF, and via other means, such as microdialysis. Candidate biomarkers need to have elevated specificity for diagnosis and prognosis. Single biomarkers are unlikely to meet this criterion, but perhaps biomarker panels or proteomic or genomic approaches will help us to have greater specificity. And additional research is needed to validate these markers in the clinical setting. So thank you, and excuse me for going over time.
Thank you, Bob.